Chapter 3: Ion Channels as Targets of Psychopharmacological Drug Action. Many important psychopharmacological drugs target ion channels. Ligand-gated ion channels as targets of psychopharmacological drug action. Membranes are decorated with all sorts of ion channels. The most important ion channels in psychopharmacology regulate calcium, sodium, chloride, and potassium. There are two major classes of ion channels. One class of ion channels is opened by neurotransmitters and goes by the names such as ligand-gated ion channels, inotropic receptors, or ion channel link receptors. The other major class of ion channel is opened by the charge or voltage across the membrane and is thus called a voltage sensitive or voltage gated ion channel. Ion channels are opened and closed by actions of neurotransmitter ligands at receptors acting as gatekeepers. When a neurotransmitter binds, it causes a conformational change in the receptor that opens the ion channel. Thus, a neurotransmitter, drug, or hormone that binds to a receptor is sometimes also called a ligand, which literally means tying. Numerous drugs not only immediately alter the flow of ions through these channels, but with a delay can also change the downstream events that result from transduction of the signal that begins at these receptors. And these include changes in gene expression and thus changes in which proteins are being synthesized and many other functions. Now these functions can range from synaptogenesis, receptor and enzyme synthesis, and downstream communication. About a fifth of psychotropic drugs currently utilized in clinical practice, including many drugs for the treatment of anxiety and insomnia such as the benzodiazepines, are known to act at these receptors. Also, drugs that act on these receptors can have an almost immediate effect. This is in direct contrast to the action of many drugs that act at G-protein linked receptors, some of which have clinical effects such as antidepressant actions that may occur with a delay because it requires initiation of changes in cellular function activated through the signal transduction cascade.